After studying this particular module, you shall be able to know what are enzymes, how they are different from the chemical catalysts, learn about the history of discovery of enzymes. You are going to learn about how enzymes recognize their substrates, know about how enzymes require cofactors and coenzymes beyond the amino acids which they are made up of. Learn how enzymes are named. Now, enzymes are basically biological catalysts. Most enzymes are proteins which possess the catalytic activity. Enzymes were discovered way back in 18th century. They recognize their specific substrate and bind to them via a cleft which is known as the substrate binding site. Once they bind in the substrate, they carry out the reaction and release the products. So for this binding, they require structural as well as the electronic complementarity with the substrate. They mediate the reactions at physiological pH and temperature at rates which are much higher than the uncatalyzed reactions. Some enzymes carry out catalysis by virtue of amino acids that they are made up of, while others would require additional cofactors and coenzymes for their full catalytic activity. Enzymes are named by 4-digit nomenclature as well as their common names. This chapter is going to discuss about the history of enzymology also. Now, enzymes are biocatalysts. The term enzyme was coined by Frederick Kuhne in 1878. Enzymes are biological catalysts that differ from chemical catalysts in 3 various ways. They work in milder reaction conditions. Remember, chemical catalysts work at extremes of pH, high temperature, while the enzymes, unlike chemical catalysts, work at physiological temperature and pH. Enzymes have higher reaction specificity. They bind to specific substrates, carry the catalytic reaction and release the specific products. Enzymes enhance the rates of reaction they catalyzed by several orders of magnitude when compared with the uncatalyzed reactions. The rates of enzyme catalyzed reactions are much higher than the reactions catalyzed by the chemical catalysts. Table here depicts how the enzymes are leading to the enhancement of the rate of reactions. We can see few examples like carboxypeptidase A. The fold enhancement of the reaction in the presence of this particular enzyme is 10 to the power 11, while in case of urease it is 10 to the power 14. So one can notice the fold enhancement and rate with and without the enzyme. While triose phosphate isomerase, this is 10 to the power 9. Now, apart from these, enzyme catalyzed reactions are well regulated. These reaction rates are not only influenced by the substrate or the product concentrations, but are also regulated by the covalent modifications of enzymes. So this is one of the points how these enzymes differ from the chemical catalyst. Most enzymes are proteinaceous in nature, but remember there are also RNA enzymes that exist in nature. We discussed in one of the previous modules about the ribozymes. Now, coming on to the history of enzymology, research work on fermentation by Joseph Lussac determined that yeast decomposes sugar into carbon dioxide and ethanol. Now, Jacob Berzelius proposed that the malt extract, which is basically the diastase, catalyzes starch hydrolysis much efficiently than the chemical catalyst H2SO4. However, in mid-19th century, Louis Pasteur was the one who proposed that fermentation can only take place in the living cells. The term enzyme was coined by W.H. Kuhne in 1878. Edward Buchner in 1897 showed that fermentation in cell-free extract, illustrating that fermentation can occur outside the living cells, as opposed to what Louis Pasteur said, that it can only happen inside the living cells. He named the enzyme responsible for fermentation of sucrose maize. In 1907, he received the Nobel Prize in Chemistry for his pioneer work on discovery of cell-free fermentation. In 1926, James Sumner gave identity to the enzymes. His work on jack bean urease, which catalyzes the hydrolysis of urea to ammonia and carbon dioxide, proved that enzymes are pure proteins. He crystallized urease. The crystals consisted entirely of proteins. But his work was not accepted till John Northrop and Moses Kunitz showed that correlation of activities of enzymes, pepsin, trypsin and chymotrypsin with the amount of the protein. In 1946, these three scientists were then awarded Nobel Prize in Chemistry. In 1963, the amino acid sequence of the first enzyme, bovine pancreatic ribonuclease A, was given. And in 1965, the first enzyme whose X-ray structure was worked out was that of hen egg white lysozyme by David Phillips. This is with regards to the history of enzymology. Now let's look at how the enzymes interact with their substrates. Now, one of the most important things is the complementarity which should exist between the substrate and the enzyme. Now, let's look at what are the kinds of complementarity. First is the geometric complementarity or the physical complementarity. Remember, van der Waals forces, hydrophobic interactions, hydrogen bonding are the non-covalent forces which drive the interactions between the substrate and the enzyme. Now, this substrate binding site in the enzyme is basically a cleft in the enzyme in which the substrate fits. This is what is termed as the geometric or the physical complementarity. That means the cleft is just structurally complementary to the substrate. We can look at this particular figure. The substrate binding site is basically a cleft where substrate can go and sit in comfortably. Now, apart from this, the amino acids that form the substrate binding site of the enzyme form attractive interaction with the substrate. Remember, one has to be positively charged, other has to be negatively charged to mediate positive interaction between the two. It cannot be negative repelling each other so that substrate and enzyme can come together. It can never happen. So this is what is known as the electronic complementarity. Now moving on to the models of substrate binding to the enzyme, there are two such proposed models which decide how substrate binding occurs on the surface of the enzyme. First is the lock and key model. It was proposed by M.L. Fisher in 1894. He proposed that both enzyme and substrate possess the geometric shapes as such that they are complementary to each other, such that substrate goes and perfectly fits in the substrate binding site of the enzyme. Just like basically the key fits into the lock, most importantly, enzyme possesses the substrate binding site even in the absence of the substrate. This is a point to be noted, that in the lock and key mechanism, it is whether or not the substrate is present, enzyme still possesses the perfect substrate binding site. We can look at this particular figure. This is the lock and key model shown for the binding of the enzyme, where the enzyme is possessing the site for the binding of the substrate. This was lock and key model. There is another model which is proposed, which is called the induced fit model. It was proposed by Daniel Koshland in 1958. He suggested that the substrate binding site is not rigid, rather, but it reshapes or molds itself after initial interaction with the substrate, so that the substrate can now fit in perfectly. Let's look at this figure. We can see that there is a cleft which is present on the surface of the enzyme, but it is not possessing the exact complementarity to the substrate. However, upon initial binding of the substrate, it makes a change, it molds itself or reshapes itself so that the substrate can be perfectly fitted in the substrate binding site. So this is how enzymes bind to their substrates. Now enzymes, apart from only working with the amino acids that they are made up of, they require certain other factors which are known as cofactors or coenzymes. Now let's understand what are these cofactors and coenzymes. Most enzymes are proteins which are essentially composed of amino acids. Few enzymes require additional chemical moieties other than the amino acids for their catalytic activity. Now these chemical moieties are termed as cofactors. For example, hexokinase requires magnesium for its catalytic activity. These cofactors can be inorganic or even the organic moieties. Now if they are organic in nature, they are better termed as coenzymes. These coenzymes derive their name from various vitamins. Few of the examples have been listed in front of you in this table. Hexokinase requires a magnesium, it is a cofactor. Carbonic anhydrase requires zinc, it is again a cofactor. Pyruvate carboxylase requires biotin. The coenzyme form is basically biocytin, so it is deriving its name from the vitamin. There is succinate dehydrogenase requires vitamin B2. So these are few of the examples illustrating that enzymes require certain helpers which can be called as cofactors. If they are inorganic in nature, they can be called as coenzymes if they are organic in nature. Some enzymes can require both metal ions and coenzymes for their activity. If the metal ion or coenzyme is tightly bound to the enzyme, it is known as the phosphatic group. So it has to be a tight association between the metal ion coenzyme with the enzyme. Then it will be called a phosphatic group. The enzyme without the phosphatic group is then called the apoenzyme, and the apoenzyme along with the phosphatic group will be called as a holoenzyme. Thus, it is the completely catalytically active form of the enzyme. So holoenzyme is basically apoenzyme plus the cofactor or the coenzyme. Now let's look at how the nomenclature and classification of enzymes is done. Enzymes are given certain common names. For example, the enzymes are named by adding suffix ase, A-S-E, to the name of the substrate or their activity. For example, hexokinase mediates phosphorylation of hexoses. Urease catalyzes the hydrolysis of urea, while RNA polymerase catalyzes the polymerization of ribonucleotides to form the RNA. So hence these hexokinase, urease or RNA polymerase, these three are basically the common names of the enzymes. If we go by the standard nomenclature, it is given by IUB-MB classification and nomenclature. Why this was introduced was, with the increasing number of enzymes, common names became less popular to avoid same names of two enzymes and one enzyme carrying different names. Hence, to avoid this confusion, International Union of Biochemistry and Molecular Biology, which is IUB-MB, adopted a systematic classification and nomenclature of enzymes. According to this particular classification, enzymes are functionally classified into 6 classes, which is 1 to 6. Class 1 basically belongs to all the oxidoreductases. Class 2 is always given to the transferases, and so on. These classes have been again subdivided into subclass and sub-subclass. Each enzyme has been allotted 2 names and 4-digit classification. This 4-digit classification is the pattern how IUB-MB adopted this nomenclature. For example, EC 3.4 17.1, where EC stands for enzyme commission. The first digit, which is 3 in this particular case, stands for the class to which the enzyme belongs, that is the hydrolase. and the second digit, which is 4 in this particular case, the subclass peptide hydrolases. The next digit, which is 17, it belongs to the subclass, which is metallocarboxypeptidase. And the fourth digit belongs to carboxypeptidase A. Thus, the 4-digit nomenclature describes carboxypeptidase A, which is EC 3.4.17.1. So, in this particular table, you can see EC1 belongs to oxidoreductases. Whensoever the catalysis is going to involve oxidation reduction reactions or the transfer of hydrogen and oxygen atoms from one moiety to the other, or even the electrons. In this particular example, you can see AH plus B bind together to form A plus BH. So basically there is a reduction of B which is taking place in this particular reaction. Another example which we can see is the transfer of oxygen to A. So in this particular case, A is getting oxidized. So under this EC1 category or the oxidoreductases, what comes in is dehydrogenases and the oxidase. The EC2 are the transferases where the functional group is transferred from one substance to the other. This group could be methyl group, acyl group, amino, or the phosphate group. So this is the reaction which can be shown for this. AB plus C gives you A plus BC. So there is a transfer of B group which has taken place from AB to C. So one of the examples of transferases is transaminase or kinase. EC3, to which hydrolases belong to, is basically the formation of two products from a substrate by hydrolysis. Hydrolysis means the water will be utilized to break a particular bond. So AB plus water is going to give you AOH, which is the hydroxyl group of water, plus BH. So lipases, amylases, peptidases, which are involved in digestion of food, they are all the examples of hydrolases. EC4 are the lyases in which the water will not be involved, but it is the non-hydrolytic addition or removal of groups from the substrate. So in this particular case, CC bond, CN bond, CO bond, or the CS bond may be cleaved. This is the reaction shown for this. Decarboxylase is one of the examples. We are well acquainted with the isomerases. They come under the category or group of EC5. So the third digit will be 5 if we are talking about the isomerases in the 4-digit nomenclature. In this particular case, the intramolecular rearrangement occurs. That means isomerization changes within the single molecule. So basically AB will be getting converted into a BA kind of a molecule, which is isomerase examples which come under this particular group. EC6 is the ligase in which joining together of two molecules by synthesis of new CO bond, CS bond, CN bond or CC bonds with simultaneous breakdown of ATP. This breakdown of ATP is very much required for the activity of the ligases. Let's look at the example. The reactions of category X plus Y plus ATP leading to formation of bonds XY plus the hydrolysis of ATP, ADP plus inorganic phosphate is one of the examples. So synthetase is one of the example which comes under this category. So under this, just to list down what is the second digit in this particular 4-digit classification, 3.1 means acting on ester bonds, 3.6 means acting on acid anhydride. So basically this is going to provide us the information how the reaction is being catalyzed. Now further on, when we move on to the subclasses, this is the list of subclasses shown here. 3.4 the third digit basically, hydrolases acting on peptide bonds. So this is hydrolases with EC3. EC 3.4 means acting on peptide bonds or the peptide hydrolases, and the third digit is going to define the kind of reaction being catalyzed. either it is a aminopeptidase, either it is a dipeptidase. Various examples are shown. If it is 3.4.25, it is threonine endopeptidase. So these are few examples to tell you what the third digit in the 4-digit nomenclature basically means. Tables here illustrates the meaning of the 4th digit in this 4-digit nomenclature, which is given by IUBMB. So EC3, again we learnt hydrolases, EC3.4. If it is peptide hydrolases, EC3.4.17. If it is metallocarboxypeptidases, again when it is the 4th digit is 1, it is going to mean carboxypeptidase A. When the 4th digit is 2, it means carboxypeptidase B, and so on. So this nomenclature basically tells you the kind of reaction being catalyzed, what kind of reaction the enzyme catalyzes, whether it is a hydrolase, whether it is a mediating a oxidation or a reduction reaction, is what governs this 4-digit nomenclature. So these are one of few examples what the second digit for oxidoreductases mean, what the second digit in case of hydrolases mean, which is EC3.1, 2, 3 and so forth, so forth. EC5 belongs to isomerases. What does EC5.1 mean? So this basically tells you a description of the subgroup of the enzymes. So we just now looked at one of the example to look at the 4-digit nomenclature of enzyme. We can look at few more examples like trypsin. Its IUBMB nomenclature is 3.4.21.4. So basically this number is going to illustrate a nomenclature like M.N.O.P, which are assigned to each enzyme, where M is going to represent the class, N is going to represent the subclass, O is going to represent the subclass, N and O basically describe the reaction which is catalyzed, and the last one in the list is the P, which is going to define the subclass which differentiates two enzymes on the basis of the substrate they use. To summarize, what we learnt in this particular module, that enzymes are biological catalysts which are proteinaceous in nature. The first enzyme which was crystallized was Jack bean nuclease by James Sumner, for which he received a Nobel Prize. The first enzyme whose primary structure was given was ribonuclease A from bovine pancreas. First enzyme whose X-ray structure was given was that of lysozyme from hen egg white. Remember, for the binding of enzyme to its substrate, both kind of complementarity is required, let it be geometric complementarity or electronic complementarity. Few enzymes can work only on the amino acids that they carry, while others require certain other factors. Either it could be a cofactor or a coenzyme for its full catalytic activity. Enzymes are commonly named, if we talk about their common names, they are commonly named by putting a suffix ase to the kind of reaction they catalyzed, or named by the 4-digit name which specifies the class, subclass and the details of reaction that they catalyzed.